Hi everyone, thank you for coming to listen to my capstone project today. My name is Miriam Stearns, and I'm going to be discussing my work of assessing the impact of European green crabs on native coastal fish using citizen science. To introduce my research, I want to discuss some information regarding citizen science and my species of interest. Citizen science is an innovative and valuable tool that leverages the power of volunteers to collect ecological data. This approach not only enables widespread monitoring of our environment, but also engages the public in scientific research. And the European green crab is an invasive species that is known for their destructive impact on ecosystems. These crabs outcompete native species for resources, they can alter habitats, and disrupt local biodiversity. So my study aimed to understand the impact of invasive European green crabs on the size of native coastal fish, focusing specifically on the staghorn sculpin due to their ecological significance. The staghorn sculpin are important for maintaining the food web as both predators and prey, and serve as indicators of habitat condition. So my research was guided by two primary questions. The first one being, can citizen science collected photos be used to evaluate changes in fish size? And the second one being, what does this information tell us about the impact of European green crabs on coastal fish populations? So to answer these questions during my internship with the Washington Sea Grant Crab Team, I focused on two key tasks. The first one being image quality assessments, where I evaluated the quality of images taken by citizen scientists. Each image was ranked 1 to 4, with 1 being excellent and 4 being unusable. This ranking helped identify common issues that could affect image quality. In Figure 2, you can see an example of an image quality of excellent. Then for fish size measurements, I used ImageJ software, which is a tool for image analysis, and I measured the sizes of staghorn sculpin from the higher quality images, which would be ones in the quality rankings of good or excellent. This data was then correlated with the abundance of European green crabs to see if there was any noticeable impact on size changes of the staghorn sculpin. For my results on photo quality and usability, you can see that in Figure 3, out of the 393 images analyzed that had fish present in the images, a majority were found to be of excellent or good quality, with 71.8% meeting this criteria. Only a small fraction, 6.3%, were deemed to be unusable. And the main factors that I found that affected image quality included turbidity, which obscures visibility, and overlapping subjects, which makes it difficult to isolate and measure individual fish. In Figure 4, you're able to see a visual example of turbidity impacting the visibility of the organisms within the bin. Then for my analysis on size variation, patterns revealed that there isn't a straightforward link between the abundance of green crabs and changes in staghorn sculpin size, but as illustrated in Figure 5, size variation tends to be more prominent in these sites with higher densities of green crab, which would be Nakata and stackpole. This suggests that while green crabs may not directly alter the average size of staghorn sculpin, their presence could influence size variability through indirect ecological interactions. From my findings, I found two key takeaways. The first one being is that the majority of the images reviewed were useful for extracting additional data on fish size, creating another data stream for the Crab Team Citizen Science program. And the second one being is that although no direct pattern was observed between green crab abundance and staghorn sculpin size, areas with more green crabs showed higher size variation. So this research highlights the critical role of citizen science in ecological monitoring and data collection. By involving volunteers, we can gather large data sets that would be difficult for researchers to collect alone. The findings of this research highlight how citizen-collected data can be used to monitor changes in fish sizes influenced by invasive species, which can provide valuable insights into ecological interactions. Looking ahead, future research should expand to include additional native species and cover a broader geographical area. This will help us gain a more comprehensive understanding of the ecological impacts of invasive species like the European green crab. Then enhancing the quality of citizen science data is also important. While only a small proportion of the images were categorized as unusable, improving the quality of images ranked as fair and unusable would enable us to extract size from a greater number of images. My suggestions for improvement include implementing training for volunteers, improving review processes, and establishing quality control measures that will help with the reliability and accuracy of the data. So I would like to extend my thanks to my site supervisor, P. Sean McDonald, my faculty advisor, Tim Essington, and Ben Rubinoff, the Crab Team's postdoctoral fellow, for their guidance and support throughout this project. I also want to thank my POE cohort, friends, and family for their encouragement and support.